down their porins. These are the gateways that bacteria have to actually allow metals, minerals, or proteins to come in. They can seal them off and antibiotics won't actually make it. They can produce inactivating enzymes like beta-lactamases. These are quite famous. And these enzymes will actually break down antibiotics. And the fourth method is actually what we call efflux pumps. And these are pumps that bacteria develop to actually pump out antibiotics and other toxins that could harm bacteria. After they do this, bacteria can communicate to each other. They can use phages, which are viruses that only target bacteria. They can also go through conjugation or transformation, where they pass on mobile genetic elements. So I can pass on a resistance gene to Liase, and then Liase can pass it on to ISBIR, and then so on and so forth. So the gene is not just restricted to one genus or one species. And then they spread globally. We travel, right? We import livestock from other places, and bacteria are not strictly affecting humans. They affect animals, they affect crops. And we ship things all around, and we travel. We saw this with COVID, how quickly it could spread. Not only because it can travel with droplets, but we move around a lot. So we can help resistant bacteria to spread from one country to another. So when we want to start doing something regarding this point, and we focus on conventional microbiology, it takes days and sometimes months to actually get the information that we need. And the interesting part is also when you're responding to an outbreak, it will take you too long to actually propose the right track and the right approach. You have to isolate the strain, you have to do biochemical identification, then antimicrobial susceptibility, then we use, for example, RAPD or PFGE, and these are all days and days of work and laborious work to actually get the information. While lately, we started using next generation sequencing, which could give us the information we need in less than 24 hours. If you have a pipeline which is developed, you can be quick in responding, and you can get all the information from 20 different experiments just by using one method. So in our lab, we use two techniques. We use the short read sequencing and the long read sequencing. I'm going to spare you the technicalities. We're just going to have a look at some of the results. We've recently been engaged with the infectious diseases division at AUBMC with a Acinetobacter baumannii outbreak, and we've collected the samples. We've actually managed to help them respond to the outbreak they are facing quite quickly, and we actually managed to map this outbreak to previous samples that have been circulating at the hospital back in 2006 and 2009. So if you have collection of isolates that are old that you can map against the new ones, you can actually check for genetic correlation between the isolates. We've also worked on Klebsiella. Klebsiella is one of the notorious pathogens that is usually understudied. And we've managed to actually spot an outbreak of a mobile genetic element in Klebsiella. It wasn't actually the bug itself, but it was a plasmid that was circulating. It originated from E. coli, went into Klebsiella, and then it circulated and it reached 9 hospitals in the country. This is yet unpublished data. We're finalizing, but you can see the with the blue lines, the plasmids that have been circulating, and it actually can affect several sequence types of Klebsiella pneumoniae, not just the 383 that the one we found. Yes. On plasmids usually, and they can pass it along. This is again the Klebsiella, and you can see samples. The genetic element of the strains, they're not the same. I haven't noticed the arrows are a bit small for you to see. So the genetic elements are not the same, but the mobile genetic elements, the ones in green, are the same sequences along several sequence types of Klebsiella. And this would not be possible to detect if we didn't use next generation sequencing. Another project we worked on with colleagues, again, at the infectious diseases department and with the ICU, the respiratory doctors, Dr. Ahmad Bouail, was the secondary bacterial infections in COVID patients. It's something that was not very well looked at during the COVID pandemic. And we've collected samples at AUB during a specific period of time. And we've actually tried to seeking to see whether we're going to observe an outbreak of some sorts. However, since AUB always likes to be special, we found something that nobody else found. And that was attributed to a specific action taken at the hospital level. They actually did not do any COVID patients in the old building. They established a new unit for the COVID, including the ICU. And what we found was during the time, we did not observe any nosocomial outbreaks in the ICU wing compared to other studies. All the other studies in the literature, they've suffered from nosocomial outbreaks, while at AUBMC, there wasn't one during the period of the study, which was the peak of the COVID pandemic. And that's attributed to the fact that the COVID critical care unit and the COVID unit of non-critically ill patients were newly established in March 2020. And they've just had a building that had nothing to do with ICU or the usual patients, and they've established that for COVID, which proved to be very smart as an outcome. After that came cholera, and in collaboration with Dr. Matar, we were testing for the whole country at the start. Cholera outbreak started in Lebanon on October 3rd, 2022. That's when we received the first sample. And one very good thing came out of using NGS was a very quick response, because we could track the strains and the Vibrio cholera that was circulating in the country in food, in water, in sewage, and in humans at a in a very efficient manner. And then we started sequencing the strains that we are getting. We discovered that the cholera strain in Lebanon belongs to the genomic wave 3 of sequence type 69, which is brilliant. And then it clusters with the strains that come from Pakistan, from Iraq, Afghanistan, and from Syria. And there is no surprise, because if we go back in time a bit, the cholera outbreak actually started in May 2022 in Karachi. Karachi was swamped. There was monsoon, there was floods, there was outbreak of acute watery diarrhea, which was 80,000 cases every 24 hours. That's quite high. In June, I was actually in Kabul with WHO. We were setting up a next generation sequencing lab for the Central Public Health Lab, and they were complaining of heavy load of acute watery diarrhea. We did a few tests. We discovered they've had a big cholera outbreak there. And then it moved to Iran, to Iraq, to Syria, then to Lebanon. And the strains cluster well together. As you can see here, they cluster very well. These are the Iraqi strains, the Pakistani strains, even the Indian strains. I mean, India, Pakistan, they share borders, so it's not surprising that they're going to cluster well together. But then we looked a bit deep, and we were finding a few strains that did not belong to that cluster. They had a bit of a different phenotype, and genetically they were not related. They were actually from the Yemeni cluster, and the Yemeni outbreak of Vibrio cholera is an XDR Vibrio cholera outbreak, which is or an MDR. It's a multi-drug resistant strain that is not easy to treat and not easy to handle. We did not find it everywhere in the country. We found it in very few locations and very few samples. So out of the 460 odd samples that we worked with, there was only 30 samples of the Yemeni-like strain cholera, which made us wonder two things. Did cholera arrive when we started looking for it, or did it arrive before? Were there these odd cases that nobody looked for? And why do we have these two strains? Why it's not one strain? We'll leave the answers for you to think about. After that, we started expanding with the cholera. So we started looking at AMR, because every day we get samples from various places, from AUBMC, from other hospitals, and we look at the percentage of MDR, XDR, and PDR. PDR stands for pan-drug resistant. We used to see one every 3-4 months. Lately, we're seeing one every day. And we're not covering all the hospitals in the country. We're not looking everywhere. And seeing one pan-drug resistant bug every day, that means one person every day in this country has no treatment. So we started doing a One Health approach, and a good few members of the team started looking at soil, sewage, water, irrigation, food. We teamed up with an NGO that deals with wild animals, not domestic animals. So we're talking animals that we didn't know exist in the countries. For example, otters. We have a flock of otters in Akkar. We didn't know they existed. They were very well protected. We sampled those otters. We got feces samples, and they actually have carbapenem-resistant E. coli in their feces. And these otters have not been in touch with humans or with the society, as far as we know. So as you can see from the map, we're doing a proper systematic, full-on One Health approach to look at the burden of antimicrobial resistance in Lebanon, looking at the whole circle. Humans, so from hospitals, we're looking at environment, and we're looking at animals. And we focused our search, because if you want to look at everything, it's going to be chaos. So we focused our search on carbapenem-resistant gram-negative bacteria, thinking that we're going to end up with a few samples, we're going to be okay. But that was not the case. All the dots you see on the map had more than one sample of carbapenem-resistant gram-negative bacteria, which also tells us that we are really struggling with antimicrobial resistance in Lebanon and globally as well. We also expanded further our work beyond Lebanon, and we teamed up with colleagues at CDC and WHO EMRO, and we got a fund by Gavi to conduct a surveillance study on XDR typhoid. So typhi in our region is in the whole EMRO region. EMRO is different than MENA. EMRO is MENA plus Pakistan, for the ones who don't know. And Pakistan have been struggling with an XDR typhi outbreak since 2017. And since travel happens quite a lot of countries suffered from this outbreak, but there was no data about this. In the last 20 years, only 10 studies were published out of Pakistan and 0 from the region regarding the XDR typhi. So we don't know what the real burden is. So we started with a literature review. We looked at what has been published. We found the gaps. And then we started with collecting samples from several countries. We started with Pakistan, Oman, Iraq, Oman is here, Iraq, and Jordan. And now we're adding the Gavi eligible countries. That's Sudan, Somalia, Afghanistan, Yemen. We're getting samples from these countries, and we're looking at XDR typhi. We've collected 106 genomes. We looked at their susceptibility, and not surprisingly at all, 98% resistance to chloramphenicol in all the strains we've seen. Ampicillin 70%, ciprofloxacin at 50% for Salmonella typhi. That's an alarm. We didn't find any colistin or meropenem resistance yet, but the sample size is small, and we haven't covered the whole grounds yet. Hopefully we don't find, but we'll see how it goes. And then we started looking at how these strains cluster with the strains that are usually worked on by the CDC, because they collect good quality data, and they can know if you've had travel history or not, especially to the region. You can see that the samples from Pakistan, they cluster quite well with the ones that come from the US with travel history to Pakistan. And this is how the strain has been spreading. This has been on the radar of several health organizations, WHO, CDC, and MSF for quite some time. And instead of just using this core genome MLST to actually type the strains, we developed a new method, which we're about to publish, and this is called the JCARD index. And the JCARD index allows us to get a lot more information. So you can see how they cluster here all the strains, and they are color-coded, how they cluster separated from each other, but then there is a good relationship between the ones from Pakistan and the ones from the US. But then if you zoom in at each cluster, you can actually tell which genomes are highly related, which ones are not. And when we say highly related, we're talking more than 99% related. We also managed to cluster them not only by genomic relatedness, we can actually look at their AMR determinants and see if those genes are related. Because a CTXM gene or an OXA gene, say OXA66, has a limit of what SNPs they can have, right? What single nucleotide polymorphisms they can have. So we can actually check if these OXAs are all related or these CTXMs are all related. And we actually managed to do this using this method. As Salim was saying in the introduction, all of this is driven by a question. Why is it always when there is war, there is more resistance? Why is it infection? Yes, there is chaos when there is war. There is lack of physicians. There is lack of microbiologists. There is lack of nurses. But there is a main reason why war always drives antimicrobial resistance in a more vicious way. And when we tried to plot based on what we see in the literature, in the 1970s, during the Vietnamese War, the first MDR strain was isolated, multi-drug resistant. In 1985, there was several wars going on worldwide. And that was when the first XDR, which is the extensively drug-resistant strain, was isolated. And then in 2007, the first pan-drug resistant strain was observed. So is there a coincidence what we are observing, or is there a real reason behind this? So we started looking at how can a bullet or a shell or destruction drive antimicrobial resistance? And why could that be a question? Heavy metals, which are made from, which constitute bullets, shells, buildings, anything that can be destroyed, previously were used as antibiotics, copper, nickel, zinc. Some of them are still used in treating certain parasites like arsenic, and they are still used in anti-cancer treatment. Antimony as well. And antimony is one of the main constituents of bullets. So, and bacteria, they, as we said at the start, they have these efflux pumps that could pump out antibiotics and anything that can kill them. So is there a relationship there that we are missing? Is there a driver that's happening that we are completely ignoring? So we started looking at conflict countries and what they are observing with Acinetobacter baumannii. And we had collaborators in Libya who sent us samples from 3 hospitals. And we tried to look at any of these strains at a genomic level to see whether there is a driver of that sort. We found that most of these strains are XDR because of efflux pumps, not because of the standard genes that we are after. So efflux pumps can be activated by heavy metals, by environmental factors, or by antibiotics. So we tried to actually figure out what was going there, but we noticed something very interesting. If you look at the figure on the lower end here, these are 2 clusters of Acinetobacter baumannii isolated from 2 different hospitals. And look how they cluster separately from each other, right? This hospital has this pathogen, and this hospital has this pathogen. And this is how you can track a patient where they are coming from. But when you look at the ones from Libya, it's a fiasco. You cannot tell which hospital is what. Every single clone is located in the 3 hospitals, and the 3 hospitals are quite far apart. They're not in the same city. There is some in Benghazi, some in Benghazi, and there is some in Tripoli. Burn patients, so these are war-wounded patients as well. It's very important to highlight this. War-wounded patients, not just any patient who walks into the hospital. And we started looking for one of these 3 things. So cross-resistance, co-resistance, or co-regulation. So cross-resistance, it's whenever one efflux pump can pump out both the antibiotic and the heavy metal, and it could be activated by either or. So co-resistance, it's whenever you have a gene which is responsible for heavy metal resistance is located on the same plasmid with a gene that's responsible for antimicrobial resistance. And this is quite common when it comes to mercury and antimony. And co-regulation is when both genes are regulated with the same regulator. This work is never ending. We're still going. We're still looking at, we're focusing on Acinetobacter baumannii for two main reasons. For the ones who don't know, Acinetobacter baumannii first was known as the Iraqi bacter. And that's because the American soldiers, when they were flying out of Baghdad after the Iraq invasion, they were being flown to Ramstein, where I was doing my postdoc. They were diagnosed with a bug that the physicians have not seen before. So they were just calling it the Iraqi bacter. And the news was full of what's this new Iraqi bacter, blah, blah, blah. After a bit of sequencing, they figured out, oh, it's Acinetobacter, and it lives in soil, it lives in water. But what they didn't continue the story to tell you that Acinetobacter baumannii is one of the strains that harbors the highest resistance determinants, and it can give them to any other genus or species. It will incorporate it very quickly to its chromosome, hence it will always be there, and then it can pass it on to others. This is what what we've been focusing on the most. And to add another factor that plays a role when it comes to conflict, we've been looking at substandard antibiotics, because whenever you have war, you have chaos, you lack doctors, you lack regulations. Exactly. You'll treat with whatever you find. Sometimes, so one of our colleagues, who's a very dear friend, Dr. Ghassan Abou Sitta, he used to go perform surgeries in Gaza or wherever there is conflict. And he used to tell us, they used to come and dump antibiotics with large quantities. And they tell you, you have 2 months to use this up before the expiry date comes in. And they would start distributing antibiotics left and right. I mean, meropenem for somebody walking into the hospital for no reason. You're coming in with a patient. You get a meropenem shot, off you go. So we started looking at these antibiotics that get dumped and the ones that get produced within the region. We looked at antibiotics that are produced in Syria versus ones that are produced in Lebanon, versus ones that are produced in Turkey, versus ones that we import. None of the ones produced in the region comply with the right dose. So if the pill says 500 milligrams, at best you have 150 milligrams, at best. So that's underdosing. Syria. Yes, Syria and Turkey. Lebanon, they stopped for a while, and then the new batch that we tested actually complies with regulations. But the problem is a lot of the refugees in our country and the refugee camps, what they use is the antibiotics that are cheaper. These are the ones that come from Syria and from Lebanon. And if you would tie this study to this slide, which talks about the One Health approach, look at the AMR genes. They are mostly closer to the refugee camps. So the whole thing is just a closed circle that has been taking us in loops. And at some point, this is going to get to all of us if we don't do something. So we haven't published yet the substandard antibiotics. We're looking for a political way to avoid a fiasco. We couldn't actually bring from Jordan yet. We're trying to get some collaborators to, we got these in a very documented manner in order to protect ourselves whenever we wanted to do a publication. So we're trying to do the same for, we're looking at Jordan, we're looking at Egypt, and we're looking at UAE, because they've also been producing quite a good bit as well. Yeah, with this, I would like to conclude, and I would like to thank collaborators that I work with, both at AUB and outside. I would like to thank CDC, Gavi, DCIF, and WHO for funds. And thank you for your attention. And I'm happy to take your questions. And sorry, I walk too much, but I cannot stand still. So
thank you for this very alarming presentation. So for our friends on Zoom, you can write your questions in the chat box, and I will read them for you. And now the floor is open for questions, if there are any.
Thank you. Okay. Now, a little comment about the drugs produced by the Lebanese company. I think that they are good.
yeah,
What's your comment, especially about the drugs produced in Lebanon?
yes, the ones that are produced in country, they
Yes.
comply with the dosage. So if it says 500 milligrams and you do an MIC and the MBC as well, you get the right dosage. But the ones that are produced outside Lebanon and the neighboring regions, they don't comply at all, which is really bad. Because with this economic crisis that we're facing, people are going for the cheaper option.
Yes, thank you. And thank you very much for mentioning Acinetobacter, our difficult bacteria to treat. Yes.
Absolutely.
One of your figures showed that colistin was not detected in your samples as
And the salmonella, yes, and the
colistin. One of our colleagues, unfortunately, he left EUB. He was at FAFS.
Dr. Asmat Qasim, yes.
Qasim has published quite a bit on colistin, and actually we formed a committee, went to Inaya as a dean, presented in front of a parliamentary committee with a lot of alarming data that has been published in, again, the One Health approach.
Yeah.
But the industry and others were saying your data is not correct.
There is colistin resistance. His data is correct. We've seen those isolates as well. There is colistin resistance. This is in Salmonella typhi. If we find colistin resistance in Salmonella typhi, we are in a lot of trouble. But what he found colistin resistance in E. coli, which we find as well. And Dr. Awar can tell you, they treat patients who regularly have E. coli or Acinetobacter or Klebsiella or Pseudomonas that is colistin resistant. And this is actually, you can find colistin in the effluents samples and in water.
Yeah, thanks. And this clarifies it. So Asmat, I remember Asmat saying it could be an indicator, because if you find colistin resistant, it means you're really at the edge of a catastrophe, because it's an antibiotic that shouldn't be used except by Dr. Awar and others in very specific cases. So do you sort of monitor that on the side?
Yes, we do. So when we're doing the One Health approach, which is here all across Lebanon, and I don't mean to disrespect any other study, but there hasn't been a single One Health approach published from Lebanon. We all say we're trying to do One Health approach. Nobody has actually took the country from north to south, looking at water, sewage, irrigation water, tap water, drinking water. Water alone is 9 categories. Then you've got soil, then you've got dust, then you have to look at animals, domestic and wild, and then you have to look at patients, and then you can say you've done a One Health approach. This is what we're trying to do, but this is not an easy task.
Just a comment about colistin. Actually, yes, I agree fully with you, and for the audience as well, is that colistin is an antibiotic. It's a little bit old, but it was like rediscovered. And why? Because we have got a lot of resistance to our current antibiotics. And about 10 years ago, actually, the first colistin-resistant isolate was reported from China, and then they discovered over there that they were feeding the pigs with colistin.
Absolutely.
For more than one purpose, for antibiotic use, maybe treating them, as well as we know that antibiotics increase the growth of animal production by increasing their weight. They will eat more, they will drink more, and other things. And so whenever we would have an organism that is resistant to colistin, it really sends an alarm, because we don't use or we don't think of colistin except at a high level of resistance. Kindly, if I may ask you about the wild otters in Ankara, where you have isolated carbapenem-resistant isolates. What do you think? How did it occur there? Those are completely in the wild. Was there a breakage in the sewage going into the water that they swim in, or what do you think, please?
So it's the water source usually, and this is what helps quite a lot of the bacteria to travel very quickly. I mean, water is a very nice transport medium for them, and through water, this is what we suggest. I mean, otters, they spend most of the time in water as well. We're trying to actually now, with the sequencing that we're doing, we can map and see whether any of the water strains that we will find matches the one in otters. Then we have the definite answer.
Hi, thank you for the presentation. I wanted to ask, how are you determining the resistance profile? So you have the sequences, is it computationally predicting resistance or wet lab experiments? Or...
We actually do both.
Okay.
We do the phenotypic and then we confirm it genotypically. Yes, lately it has been proven by, not only by us, by several other people, that the genotype will always match the phenotype. But the efflux pumps, they are the tricky part. That's why we always do the phenotype and then we look for the genotypes to confirm. Dr. Adib.
Thank you very much for a very nice presentation. I have two comments actually. The first one is to sort of give you the good news that we're starting to think about One Health surveillance. The epidemiological surveillance unit at the Ministry of Public Health has recently been forced to start monitoring sources of water for cholera.
We're doing the testing for them, so.
Yeah, so the idea is working, it's spreading, and then recently there was a common meeting that included the folks at the veterinarian surveillance, and it's when you realize that there might be 15 different employees, but there's only one who can read and write, and that's a very serious problem. But they are everybody is warming up to the idea, and I think we need to accelerate it because definitely we have it's not just this problem of surveillance that is a problem, it's also spreading of zoonosis that will probably cause the next pandemics as we go forward. But it's important that we all light up the light wherever we are and say you need to do it now, otherwise it won't. My second comment is and I need your opinion here. Sometimes when you do not look, you don't find.
Yes.
And so when you look and you find something, you have a tendency to sort of link it to what's happening around it in the context. So we found it in 78, so it must be Vietnam. Whatever. But it may actually have been going on for quite a while, and the carbapem resistance in otters and the wild animals in Lebanon, we don't know when it started. It did not start when we discovered it.
No.
How would you know when it started?
So I think there is no right answer for your question. And unfortunately, it's not simple. We cannot do carbon-13 dating and we'll say this is that old and this is newer. But the idea is it was the first reported, that's the right terminology, from an American soldier who was fighting in Vietnam and who was injured in Vietnam. This was the first reported MDR. Could we have looked for it? Yes, before. Could we have found it? My answer is maybe. If you look at GLASS, which is the global surveillance for antimicrobial resistance that WHO runs, not all countries report. And some of the very well-developed countries, they don't report. I mean, GCC countries, not all of them report.
It's really very much hit by the fact that those that are reporting most are actually underdeveloped.
Absolutely. Because these are the ones that are more eager to be seen somewhere. So I do work with WHO on establishing labs for next generation sequencing in the complex emergency countries. So I only go to Afghanistan, Somalia, Sudan, Yemen, Libya, Iraq, Syria, and Lebanon is now unfortunately included on the list. And the idea is we see how eager these people are to learn and once they get this right training and this opportunity, they would actually put themselves on the map. These countries are now sequencing COVID in-house and they are reporting their own data on GISAID. Given the right training, they will get into AMR as well.
Thank you for a very well talk.
Thank you.
How often Acinetobacter infects patients, and then what are the symptoms to alert the doctor to know that there is an Acinetobacter?
I think maybe you can answer better. You're more qualified, Dr. Abou.
Thank you. Acinetobacter is a typical nosocomial pathogen that occurs in the hospital, especially in ICU patients or in patients who need more advanced medical care, with the tubes, with catheters, with the drains, surgeries. Those are the patients who would have the infection. And there is no clue that this is Acinetobacter unless it is cultured or sequenced and we know about its antibiogram or the resistant pattern.
Any sample. There is no best sample.
Any sample. For a febrile patient, we should not forget blood cultures are very important, and it depends on what we suspect. Respiratory, if it is in the chest, urinary, abdominal, post-surgical, it depends on the case. A blood sample is very valuable in case of fever, because in case it's positive, it would be very helpful. And those patients would have central venous catheters. So a blood sample is always very important with fever.
You have to have the time. You have to have the time.
We don't always have sequencing.
We're trying, maybe in the near future, it could become a clinical test, which will give information much quicker.
So again, very interesting. So a lot of questions could come up. So I have two questions related. They're into the realm of politics and policy. Okay. So when you do One Health, you cannot think of One Health without thinking about the politics and the context of a country. So Lebanon is not an island. And so how can you look at the data in the context of Syria, across the borders, or the origins there? So what are we doing there? Because I think we cannot control this without some kind of this scientific interaction. The second question relates, how optimistic are you about actually winning this war against AMR?
So answering the first question, I'm going to answer it with an example, a real life example that happened to us. Even if we would have closed the borders when the cholera outbreak started in Syria, we would have still received cholera, not because of smuggling, but there is water rivers that flow all the way from Iraq to Lebanon, and Vibrio cholera loves water. And they will tell you, oh, it goes down the sea, it will die. It actually loves salt. So salty water is a more preferred environment. So Lebanon is not an island, but we need to start doing something. First of all, the first very important thing we need to do is to implement prescription policy for antibiotics. The amount of antibiotic consumption since the start of the pandemic in Lebanon until last year was 3 times more, according to the Ministry of Health and Customs website, is 3 times more the last 10 years. And we're talking 3 years versus 10 years. During the 3 years, it's 3 times more to treat what? A viral infection with Zithromax and ciprofloxacin. So we need to implement prescription policy. That's step number 1. Step number 2, the right people need to sit down and speak with specifically what Dr. Adib was mentioning, the veterinary, poultry, aquaculture, all of these people, they need to understand you cannot just feed colistin, as Dr. Awar said. Colistin is it's a big peptide. It's a growth hormone, and they give it as prophylaxis. Same as tetracycline. You cannot just feed them to the chicken. I mean, there are countries who actually did something. Look at the numbers in Denmark, in Netherlands, in Belgium, in Sweden, in Finland. Their antibiotic consumption dropped by 80%, and they actually can control antimicrobial resistance. They can do something. Are we in Lebanon gonna manage to win this war? I'd rather not answer your question. I'm gonna speak globally, because with the current people in charge, I doubt it. But worldwide, there is a lot of work that needs to be done. You need new antibiotics.
Advocacy is what Asma was very interesting of pushing he was fought back quite.
Yeah.
But there's an issue of advocacy that physicians, basic scientists, public health people, vets, all the others can perhaps mobilize this beyond colistin to talk about this issue. I don't know whether Salim,
Once
you're
we
following.
work all as a team to advocate for this problem, this is when you will start observing. But when I want to fight for my own piece of cake and somebody else for their own piece of cake, we'll
As a
never have a
scientist, now
message.
you're talking.
Yes.
Yeah.
So to win this war on antimicrobial resistance, you need new classes of antibiotics. We don't need new antibiotics. So we don't need a new box of meropenem plus some inhibitor. No, we need new classes. And yeah, there needs to be a lot more research in antibiotic drug discovery, which is another part that we work on. But we as a small lab will not be able to solve the problem alone. You need the big companies to get into that. Where the money is, where the capabilities is to discover new antibiotics in order to try and do something. And you need to look at the big countries. I mean, Lebanon, we're what, 3 million, 4 million? You need to look at China, India, Pakistan, Afghanistan. You need to look at a 3 billion population that could be restricted to 5 or 6 countries. And these are the ones who actually need to start doing something about it.
Said that we need new big companies to look at that.
We need big
That's
companies,
what-
yes, absolutely.
And big-
Stop. Really?
I have a question, maybe related to people who are infected with drug-resisting bacteria. Is there any solution for them, any treatment for them, or any possibility for them to survive?
So usually we try to help in an in vitro test. We do something called a checkerboard assay, where you can combine 2, 3, 4 antibiotics and see if they're effective. But yet again, this is in vitro. We cannot really translate in vitro data to in vivo data without the proper method. And combination of antibiotics will differ even if you have the same bacterium, but different resistance mechanisms, the combinations will be different. And that's in vitro again. That's why we need new antibiotics, but we need the antibiotics to be used in a smart way, like rifampicin. Rifampicin is being protected worldwide for TB. So we need to get more antibiotics that are of new classes. There are a few in the pipeline, but it's taking ages. And just between parentheses, if all these antibiotics that are now used in the clinic would go back to step 0 in FDA, 1 will pass and all will fail. All. Colistin is nephrotoxic. Tetracyclines, same thing. Carbapenem at high doses, it's toxic, right? So now
What's
the
FDA
rules
doing
are
then?
more difficult to actually get an antibiotic into the clinics. It's more difficult. So yes, they have to look at the rules from a wider angle, and we need to find new antibiotics that can actually work.
That by that, it will be expensive because a lot of investment.
Absolutely.
You have to find replacements that are cheaper and the cycle. That's the big challenge of this.
This is one of the reasons why the big pharma stopped looking into antibiotics. It's a short course treatment, reward is very little, and to find them, it's very expensive.
Has to be a social justice case.
That's why.
And now I see your point. Yes.
Also, maybe just to remind of the One Health concept is that clinicians, and we doctors, we are using the smaller portion of the consumption
Absolutely.
of antibiotics. Okay, it's used much more in chicken production, in poultry, in animal husbandry, animal production,
And household as well.
in fisheries, in everywhere.
Yeah.
everywhere. So it's a One Health approach, surely.
What's really familiar to me is what I hear from hospital and the outbreak of Acinetobacter. And so we're talking about more controlling the antibiotic, but what's happening when we have an outbreak and to what extent hospital are being able to control the outbreak when it happens. And we have really good mechanism or good control mechanism, because it's really, I know, and we've heard it, it's not out, but we hear about really increased number of death because of this
Drug-resistant Acinetobacter. So I mean, how well can you prevent it? At the end of the day, this is a bug that lives in soil and water, on your clothes, on your gloves, on AC system, anything. So that's why there is so little you can do, or limited factors you can do to try and control. But the idea is to respond faster. And I think if you would speak with the infection control team at UBMC, they would tell you that they are getting faster with every outbreak, regardless of sequencing. They are getting faster and faster because now they know what to do. With sequencing, they get better info. No, the personnel are getting faster, but the outbreaks are also getting more vicious, not only faster.
Yes.
The thing is, this is what I know at AUB. I would rather not talk about other hospitals, although we work with them, especially when they have outbreaks, but usually they don't disclose that they have outbreaks.
Just let me say that Acinetobacter has forced closure of the ICU
Yes.
for a couple of weeks, transfer it to somewhere else, clean up everything, and then go back.
It was during this outbreak.
And this has been repeated over and over. Yes, it closes our ICU.
My information is like about 10 years ago. This is when I was in the States. There emerged highly resistant Staph aureus and some strains of streptococci. All what they could have done, and they have done, is put those people in isolation. This is for protection, but still the Staph aureus and then those certain strains of streptococci were already spread through so many things like traveling and then mixing with etc. So that's all what they have to do to protect it, put them in isolation and leave everything up to chance and God's will.
Yeah, one of the things that we always forget about is bacteria and viruses are completely two different creatures. Viruses cannot multiply on their own. They cannot live on their own. Bacteria, they can do all of that on their own. So one of the other reasons, so viruses are more known to spread via air droplets and can travel further because they are lighter. Bacteria can multiply more on their own, and they live longer. So these are the, if the creator would have put all of them in one, we know we're fighting one monster. But yeah, exactly.
Okay, with that, we come to the end of this seminar. Thank you, Dr. Antoine, for this very eye-opening info. Thank you, everyone.